Hello everyone, welcome to our demonstration of Sds Page, which stands for Sodium Dodecyl Sulfate Polyacrylamide Gel Electrophoresis. Sds Page is one of the most widely used analytical techniques for separating proteins based on their molecular weight. It is an essential method in biochemistry, molecular biology, biotechnology, and medical research. Scientists use Sds Page to determine protein size, evaluate protein purity, compare protein expression, and prepare proteins for further analysis, such as Western blotting or mass spectrometry. In this video, we will demonstrate the complete Sds Page procedure, explain the scientific principles behind each step, and show how to interpret the final results. To understand Sds Page, we first need to understand how proteins behave. Proteins naturally differ in size, shape, and electrical charge. These differences would affect how they move through a gel. Therefore, before electrophoresis, proteins are treated with sodium dodecyl sulfate, or SDS. SDS is an anionic detergent that denatures proteins by disrupting their three-dimensional structure. It coats the proteins with a uniform negative charge, making the charge-to-mass ratio nearly identical for all proteins. As a result, proteins are separated primarily according to their molecular weight rather than their shape or native charge. When an electrical field is applied, the negatively charged proteins migrate toward positive electrode. The gel contains two layers. The stacking gel has larger pores and a lower pH. It compresses all proteins into a thin, concentrated band before separation begins. The resolving gel has smaller pores and a higher acrylamide concentration. Here, proteins separate according to size, with smaller proteins moving faster and farther than larger proteins. A protein ladder, also known as a molecular weight marker, is loaded alongside the samples. It contains proteins of known molecular weights and serves as a reference for estimating the sizes of unknown proteins. The electrophoresis buffer conducts electricity, maintains the correct pH, and facilitates efficient protein migration throughout the experiment.
Firstly, clean glass plates are assembled through spaces to form a lead broadcasting chamber. The resolving gel solution containing acrylamide, trace HCl buffer with pH 8.8, SDS, ammonium persulfate APS, and TMAD is then prepared. APS and TMAD are added last as they initiate the polymerization reaction. The resolving gel solution is poured between the glass plates and a thin layer of distilled water is carefully added over the gel surface to prevent oxygen inhibition during polymerization and to create a smooth level interface. The resolving gel is then allowed to polymerize for 45 minutes. After the resolving gel has polymerized, the stacking gel solution is prepared using acrylamide, trace HCl buffer with pH 6.8, SDS, APS, and TMAD. The distilled water is then drained from the surface of the resolving gel and the comb is prepared. The stacking gel solution is poured on top of the resolving gel and the comb is carefully inserted to create wells for loading protein samples. The stacking gel is allowed to polymerize completely for 35 minutes. Once polymerization is complete, the gel is ready for use.
Before loading the samples into the gel, the protein samples are prepared by mixing them with Lambda sample buffer. This buffer contains SDS, a reducing agent such as bemethactoethanol or DDT, glycerol, and bromophenol blue. SDS nature the protein and give them a uniform negative charge, while the reducing agent break this sulfide bond to fully unfold the proteins. Glycerol help the sample sink into the wells, and bromophenol blue act as a tracking dye during electrophoresis. After mixing the sample, are briefly centrifuged to collect the solution at the bottom of the tube. The samples are then heated at 95 Celsius for about 5 minutes to completely denature the protein before being loaded onto the Sds Page gel. After the protein samples have been prepared, the next step is sample loading. First, a protein ladder is loaded into the first well. The protein ladder contains protein of known molecular weight and serves as a reference for estimating the size of the sample protein. Next, the prepared protein samples are carefully loaded into the remaining wells using a micropipette. The pipette tip is placed just inside each well and the sample is dispensed slowly. to avoid damaging the gel or introducing air bubbles. Carefully loading is important because it ensures accurate protein separation during electrophoresis. Once all the sample has been loaded, the gel is ready for electrophoresis where the protein will migrate through the polyacrylamide gel and separate according to their molecular weight. After all samples have been loaded, the gel chamber is closed by placing the lid securely onto the electrophoresis tank. The electrodes are then connected to the power supply by matching the correct terminals, with black connected to black and red connected to red. The power supply is switched on and the voltage and the timer are adjusted according to the experimental requirement. During electrophoresis, bubbles can be observed at the electrode due to electrolysis of water, which indicate that an electric current is flowing through the chamber. At the positive electrode, oxygen gas is produced, while hydrogen gas is produced at the negative electrode. The electric field causes the negatively charged protein coated with SDS to migrate toward the positive electrode. SDS give each protein a similar negative charge to mass ratio, allow separation mainly based on molecular weight. As the protein moves through the polyacrylamide gel, the gel acts as a molecular sieve. Smaller protein can pass through the pores more easily, allowing them to migrate faster and further while larger protein move more slowly. After the electrophoresis run is completed, the separated protein bands can be visualized through staining or transferred to a membrane for further analysis, such as Western blotting.
Moving on to the this image shows us the gel image, and no one is our molecular weight marker, which serves as our ruler, and then to add on to the sample. And by looking at the image, we can clearly see that there are two distinct prominent bands on link 2 and link 3. By comparing them against our marker, we can estimate their molecular weight around 55 kilodalton and 35 kilodalton. While Sds Page can tell us about the size and the purity of the protein, it cannot definitely tell us their exact identity. So, to confirm it, we need to do a validation technique, which is using extend blotting and mass spectrometry. Now, we will go to the first method for validation, which is Western blotting. So, while the gel shows us all the protein of certain size, Western blotting lets us hunt for our specific protein, just using purines. So, we will transfer the separated proteins from gel into a standard membrane. Then, we will block the membrane to prevent background noise using photo and incubate it with primary antibody design specifically to bind to our target protein. So, then a secondary antibody conjugated to enzyme or fluorophore is added. Then, it will allow us to detect and visualize the signal if a band direction and our perspective. As the molecular weight, it confirms the presence of our specific target protein. And next, we will use the mass spectrometry. So, for this process, we will physically excise the 55 kilodalton and 35 kilodalton band that directly from our stain Sds Page gel. And we will digest protein that inside the gel matrix into the smaller pieces called peptide, usually using an enzyme reaction. This peptide then vaporize and ionize inside the mass spectrometer. The instrument measures their precise mass to charge ratio. Then finally, the software will compare the mass measurement against the massive genomic database. Then finally, it will give us the exact amino acid sequence and definitive proof of the protein identity, converting our workflow from simple preparation to absolute identification. So from the result, we can confirm that the Sds Page gel successfully separate the protein sample to 2 kg and approximately 55 kilodalton and 35 kilodalton. While this tell us the size and relative purity of the protein, such alone is not enough to prove their exact identity. So to confirm this protein, we must use Western blotting to allow us targeting and identifying the protein using specific antibodies and using our mass spectrometry to provide a definitive high-resolution result of their amino acid sequence. So by combining these three methods, a basic visual inspection turns into a highly accurate one and confirm the identification of the target protein.
In conclusion, Sds Page is a simple yet powerful technique for separating proteins according to their molecular weight. By denaturing proteins with SDS and separating them through a polyacrylamide gel under an electric field, researchers can accurately analyze protein size and purity. Sds Page remains an essential technique in analytical biochemistry and serves as the foundation for advanced methods such as Western blotting and mass spectrometry. Thank you for watching our demonstration.